Welcome to the AMDA webinar BMN-701, Potentially More Effective Treatment Option for Pompe. Your speakers for today's program are Barbara Wuebbels and Jon Lebowitz. Jonathan Lebowitz was the Executive Vice President and Chief Scientific Officer of ZyStor Therapeutics, where he was responsible for the development of Pompe candidate therapeutic ZC-701, now called BMN-701. Dr. Lebowitz is an accomplished investigator with over 25 years of research experience in the biochemistry, molecular, and cell biology of organisms ranging from microbes to humans. Barbara Wuebbels has been with BioMarin for the past 3 years and is currently Manager of Medical Affairs, Investigator Relations, where she works closely with patient support groups, national and international rare disease organizations, and also serves as an in-house consultant for various project teams. Ms. Wuebbels has worked in the world of orphan drugs and rare diseases for the past 11 years. Operator assistance is available at any time during this conference by pressing £0. A question and answer session will follow the presentation. You may send chat questions at any time during the presentation using the chat window indicated by a speech bubble icon located to the right of the presentation. I will now turn the call over to our speaker. Jon, please begin.
Thank you, Leo. And now I have control. Excellent. Well, thank you very much. I know Barbara would join me in expressing our gratitude to have the opportunity to speak with all of you tonight about BMN-701, which is a therapeutic that is now advancing to the clinic for the treatment of Pompe disease. I think I presented the ZyStor story at an AMDA telecon maybe a year and a half ago. And so there have been some significant advances in the 701 program, not the least of which is a change in name of the compound. But in that last year and a half, we've been able to open an IND, procure clinical supply enough to start a clinical study, and ultimately we were able to actually match what we think is a very good compound with a company that has the expertise and resources to actually see it through the clinical process, and that is BioMarin. So as I'm sure you're all aware, in August of 2010, BioMarin acquired ZyStor Therapeutics, and in particular the ZC-701 program. Now, I'm going to try very hard to refer to this compound as BMN-701, but odds are very high that I will slip up and refer to it as ZC-701 along the way, but I want you to know that we're speaking about a single compound that's simply undergone a name change in concert with the acquisition. So what I'd like to do is spend a few minutes going over the rationale for developing a second-generation Pompe therapeutic, take you through some of the data relating to BMN-701, and then after that, Barbara is going to speak on the subject that I'm sure you are most keenly interested in, which is the clinical program and where we are and when we're starting and so on and so forth. So in this slide, I wanted to make a couple of points, most all of which I'm sure you're fully aware of. First, of course, Pompe disease is a genetic disorder caused by a deficiency of the enzyme acid alpha glucosidase, which we refer to as GAA. This is an enzyme that degrades glycogen in a particular cellular compartment called the lysosome. Now, the root cause, the molecular cause of Pompe disease then is actually the accumulation of glycogen in the lysosome of cells, particularly skeletal muscle and heart tissue. And this image on the right-hand side of this slide, which comes from a paper by Beth Thurberg et al., who's a pathologist at Genzyme, or at least was at the time this paper was published, shows sort of various levels of cellular pathology in muscle tissue. And really the point not is to get into a detailed discussion of muscle physiology, but rather to point out that when glycogen accumulates in the lysosomes, many secondary effects happen that ultimately can result in the destruction of the muscle tissue. So, as I've just said, you can destroy muscle function and muscle fibers from the accumulation of glycogen in the lysosome. And then the goal of enzyme replacement therapies such as myozyme and lumazyme and our compound BMN-701 is to restore the missing enzyme, that is GAA, in the lysosome in sufficient quantities so that you can reverse the molecular defect, that is accumulation of glycogen. And then the hope is that by doing so, you can minimize the clinical manifestations of Pompe disease. Now, this slide makes the point that a eukaryotic cell is a very complicated entity. There are multiple compartments, different compartments, each of which is encased in a membrane that provides some kind of a barrier from the free movement of molecules, and particularly macromolecules, from one compartment to the other. So the objective of enzyme replacement therapy is to deliver this molecule, GAA, human GAA, shown here by this little icon, to the lysosome. Now, in this schematic, you see a single lysosome. There are many lysosomes in the cell. But the point is that to get an effective therapy, this molecule has to go here. It doesn't do any good to go into the many other compartments that are part of the cell. And so how is this accomplished? Well, most of the enzyme replacement therapies for lysosomal storage diseases are delivered to the lysosome not by directly jumping from the exterior of the cell to the lysosome, but rather in a process that is mediated or carried out by another protein molecule called a receptor. And it's a receptor because it receives molecules and then either transmits a signal or physically can move molecules from place to place. So in this case, we're talking about a receptor called the mannose-6-phosphate receptor, abbreviated M6P. So this green blob here is the mannose-6-phosphate receptor, and what happens is when you add or when a cell comes in contact with GAA that's on the cell surface, if the GAA molecule has an oligosaccharide, that is a polymer of sugars that contains mannose-6-phosphate, then this molecule can bind to the receptor, and then through a process called receptor-mediated endocytosis, you get an invagination of the plasma membrane, and this invagination becomes a vesicle, which then permits the GAA molecule to be carried through a series of different vesicles, ultimately to a compartment called the late endosome, where the pH is acidified, and this enables the protein drug GAA to get to the lysosome ultimately. So the point here is that this is a very specific process, and the only way it works is if the drug molecule GAA can bind to this receptor on the exterior of the cell. Now, this slide actually shows the rest of this pathway, and this mannose-6-phosphate receptor pathway is actually the pathway that's used to deliver newly synthesized proteins to the lysosome of cells. So in humans, animals that don't have a defect in the GAA protein. The GAA protein, a good copy of the protein is synthesized in the endoplasmic reticulum, and then it is transported via interactions with the mannose-6-phosphate receptor from the Golgi through this vesicle pathway to the lysosome. And by the way, I don't know if this slide looks familiar to you, but I think you can see it out of focus slightly behind Harrison Ford as he describes his technology in the movie Extraordinary Measures. So for these enzyme replacement therapies to be effective, you have to deliver the protein to the lysosome, and that means you have to interact with a receptor like the mannose-6-phosphate receptor. And so if you can get efficient delivery of a molecule to the lysosome, you can have an effective drug. Now, we know that myozyme or Lumazyme is a life-saving therapeutic, that in the infant onset population, for example, that infants that would otherwise die without the access to myozyme, these patients can live for many years, some of them, and this is primarily due to reversal of the pathology associated with the heart. What we know from animal studies with compounds like myozyme, as is shown on the right-hand side of this slide here, is that if you look at the reversal of glycogen storage in, say, the Pompe mouse model, there's actually a dose response that goes all the way up to 100 milligrams per kilogram. Now, the approved dose of myozyme is 20 milligrams per kilogram. In this particular experiment, which was published by Genzyme scientists, this dose actually shows very little difference from vehicle. That is, a buffer control. Now, this isn't always the case. Often you see a much better result, but in this particular experiment, this is what it looks like. So if you could deliver to patients 100 milligrams per kilogram of myozyme, you'd have a better drug. But unfortunately, that can't be done. I mean, you are likely to experience kidney problems with that high a dose of protein drug delivered into the bloodstream. And in fact, in the infant onset clinical trial study number 1602 that was done by Genzyme, at a dose of 40 mg per kg in comparison with 20 mg per kg, you see more infusion-associated reactions and higher antibody titers. So it's not possible really to give the optimal dose of myozyme. So why does it take such a high dose to get myozyme to be fully efficient at clearing glycogen from muscle tissue? Well, as we discussed before, in order for this drug to be delivered to the lysosome, you need to bind the mannose-6-phosphate receptor. And while I was referring to mannose-6-phosphate as the molecule that enables the protein to bind the receptor, you actually need an oligosaccharide, such as is shown here, an oligosaccharide with 2 mannose-6-phosphates, as indicated by these two P's. So each one of these little circles and squares is a different sugar molecule joined together. This material altogether is an oligosaccharide, and we would say this is a bis mannose-6-phosphorylated or BPM bisphosphorylated oligosaccharide. Well, it turns out that if you look at the oligosaccharides in myozyme, only 4% of the oligosaccharides have this bis mannose-6-phosphate, and that means that as is shown in this panel, if you actually test for how much of the molecule can bind to the mannose-6-phosphate receptor, this small fraction out of the total material, this is all stuff that doesn't bind, this is stuff that does bind. So approximately somewhere around 80% of the myozyme molecules don't bind to the receptor with high affinity and are not delivered. So this would be as if you went to mail your holiday cards to all of your friends and family, and you went to the mailbox, and only 2 out of your 10 holiday cards had a stamp on it. So, only 2 of those cards are going to get delivered. The other 8 cards are going to end up in the dead letter office. And it's sort of the same thing here. When you give, a dose of 20 mg per kg of Myozyme, really only, say, 4 mg per kg of Myozyme is capable of being targeted with high affinity to the mannose-6-phosphate receptor and therefore delivered to the lysosome. Now, this is actually not any longer a controversial point, and I think the best evidence for that is the fact that everybody who has worked in the Pompe area has spent an enormous effort trying to find ways to improve the ability of the GAA molecule to bind to the mannose-6-phosphate receptor. So the one example is the Novozyme technology, in which people make the recombinant GAA and then add the mannose-6-phosphate later in a test tube. This is the technology that was the subject of the movie Extraordinary Measures, and it comes from Bill Canfield's work. Genzyme has a second-generation Pompe therapeutic in development that they refer to as Neo-GAA. In this case, they take essentially Myozyme, and then they chemically couple to it a synthetic oligosaccharide that is a sugar polymer that has 2 mannose-6-phosphates on it. They chemically couple this to Myozyme, and that gives you Neo-GAA. And at BioMarin, in fact, there was a second-generation program referred to as BMN-701, in which mutant CHO cells, that's Chinese hamster ovary cells, these are sort of the workhorse mammalian cells of the protein therapeutic world. So mutant CHO cells that secrete GAA molecules with more bis mannose-6-phosphate. So each of these efforts were designed to improve binding to the receptor by modifying the carbohydrate or sugars by adding more mannose-6-phosphate. At ZyStor, we developed what we refer to as glycosylation independent lysosomal targeting. We call it that because the word glycosylation refers to the addition of these polymers of sugars to protein molecules. So our technology, glycosylation independent lysosomal targeting, or GILT technology, uses a peptide tag that is protein to bind to, in fact, the mannose-6-phosphate receptor. And our peptide tag is derived from human IGF-2, or insulin-like growth factor 2, and the reason we chose that as the basis for our targeting functional group is because this receptor, shown in schematic here, which is the mannose-6-phosphate receptor, not only does it bind mannose-6-phosphate containing oligosaccharides in these particular regions of the protein, but there's also a very high affinity binding site for IGF-2. And so the affinity of IGF-2 for this receptor is as good, or in fact better than, tighter than the affinity of a bis mannose-6-phosphorylated oligosaccharide for the receptor. So this gives us, in the case of a Pompe therapeutic, a big advantage because, as I said before, in contrast to the case where you're only putting a stamp on 2 of your 10 envelopes and then dropping them in the mailbox, we're putting a stamp on every single enzyme molecule, every single envelope. So all of the molecules ought to be delivered to the lysosome. Now, this is pushing the analogy, but we're not talking about going to FedEx and using a different delivery pathway. We're using the exact same delivery pathway, that is the mannose-6-phosphate receptor. We're just putting a new stamp on every single enzyme molecule. And so this we think gives us an opportunity to deliver enzyme more efficiently to the lysosome. So here is a very simplified schematic of BMN-701, formerly known as ZC-701. It has the GILT tag and then the GAA molecule fused together. So this is a single polypeptide, a single protein molecule. It comes out of the cell that produces it, and it's a CHO cell that's producing this molecule, intact as a single molecule. So, as I've said, our belief is that by putting a high-affinity ligand for the receptor on every single enzyme molecule, we should see much more efficient delivery of this molecule into the lysosome of target cells. And this experiment shows that we see exactly that. So this is an experiment where we take muscle precursor cells grown in a Petri plate, and we add different amounts of drug, ZC-701 or BMN-701, myozyme, or a recombinant human GAA that we actually produced at ZyStor. Different concentrations, as you can see on the x-axis. And after an incubation, we ask how much of this protein is then taken up into the cells. And what you can see by looking at the slope of the initial part of these curves, this slope for ZC-701 is much, much steeper than the slope for these molecules. And that means that the efficiency of uptake into cells is much, much greater. In fact, by this kinetic parameter we call K uptake, it's almost 30-fold more efficient. That means you, at a much, much lower concentration of protein exposed to the cells, you can deliver more protein into the cells. Now, there's actually something else interesting that can be gleaned from this experiment, and that is not only is the slope steeper, but actually at saturation, you actually saturate at a much higher level of uptake into the cells. And we actually think that this could be biologically significant, that there's evidence in the literature, in fact, that molecules like IGF-2, remember that's insulin-like growth factor 2, and that's the molecule that our tag is derived from. So molecules like this are actually capable of causing redistribution of the mannose-6-phosphate receptor so that more of it is on the cell surface. So we think it's possible, and we're certainly going to look at this experimentally, that this is what is going on here. So we've said that we put a high-affinity tag on every single enzyme molecule. That means we're putting a stamp on every envelope, and that means we can deliver enzyme more efficiently to cells. So then the question is, what about in animals? Can we deliver enzyme more efficiently to critical tissues, skeletal muscle tissue, and remove the glycogen storage? And that kind of data is shown on this slide. This is a composite slide that compares data for myozyme to data for BMN or 701. And what you can see in each of these tissues, heart, diaphragm, and quadricep, is that essentially, at a 5-fold lower dose, you can get the same level of glycogen clearance. So I should point out to you that the y-axis here is the amount of glycogen that's remaining. So the lower this number, the better. So, in this case, 5-fold difference here, 20 mg per kg is about the same as 100 mg per kg. Same thing here. So what we've shown you is basically that the logic of this compound works. That is, if you put a stamp on every envelope, that you will deliver your letter to the appropriate place. And in the case of ZC-701 or BMN-701, you'll clear the glycogen storage in critical tissues. So some of the big news for us since the last time we spoke, we in the spring of 2009, we completed our IND-enabling toxicology. Really, the only findings that we observed in tox studies were transient hypoglycemia. That means lowering of blood sugar at very, very high doses of drug, well over 100 milligrams per kilogram. That is 125 mg per kg. We filed an IND in the summer of 2009. It's now open. We have orphan designation. We have clinical material available. And finally, we are almost ready to start the clinical trial. We expect to be starting by Q1 of next year, if not sooner. So with that, I want to turn over the conversation to Barbara, who's going to describe the outline of the clinical protocol, if I can manage this feat correctly. Barbara, you should have the controls now.
Yeah, there they are. Okay, so the only thing I need to know is how to advance the slide.
There should be an arrow at the top, just to the left of the pointer.
Got it. Sorry. Okay. So the first of all, I'm delighted to be part of this conversation tonight. I am new to the Pompe world, but I'm very excited to be in it. And I did have the opportunity to meet some of you at the Pompe meeting in LA a few weeks ago, and I've been in email and voicemail contact with many of you since then. So the first study I'm going to talk about is the August 2009 clinical design study, which if you go to clinicaltrials.gov, this is the study that is posted. It's an open label study, meaning that there's no placebo involved. We are going to enroll potentially 10 late onset patients. They will receive medication biweekly for 12 weeks. The first group of 3 patients will receive 5 milligrams at each one of their doses over the 12-week period. And the way the study will work is that patient number 1 will be enrolled in the study, and after they have received their second dose, we will draw some labs. We will do a safety evaluation to make sure they're not having any kind of an infusion-associated reaction, and if they are okay, then we will enroll patient number 2. And then when they have completed 2 doses, we will do the same evaluation for them, and if they're okay, we'll enroll patient number 3. And that's the way the study will roll out. So our primary endpoints are safety, and then also looking for antibody responses. And our secondary endpoint is looking at the pharmacokinetics. In addition to that, we're doing some exploratory endpoints, which is looking at other things to see how the drug may influence them, such as GAA levels, such as the respiratory status, the forced vital capacity, which is the maximum amount of air you can breathe in or breathe out at one time. The 6-minute walk test. Can you walk at least 130 feet in a 6-minute period? The inclusion criteria for this study is patients that are from 13 to 50, and they have to have less than 75% of the lower limit of normal of GAA levels and greater than 40% forced vital capacity. They also need to be naive to GAA enzyme replacement therapy. Now, it says here, or no GAA enzyme replacement therapy for a month prior to screening. We are changing that to just naive patients. This is the study that is just about ready to go. The first site will be at Dr. Barry Burns at the University of Florida in Gainesville, and that will start in the next couple of weeks. Since that time, we have amended the clinical design protocol. And the reason that we have done that is with the acquisition of ZyStor by BioMarin, we have more capability to do a better, larger study the first time out. As you know, ZyStor was a very small company and had somewhat limited resources. BioMarin is a bigger company. We have the ability to do more study at one time. So this study will take place in the United States, the United Kingdom, and in the EU. And we are looking for approximately 30 late-onset patients. They will also receive biweekly administration of drug. But instead of a 12-week study, as was in the first design, they will receive drug for 24 weeks. Plus, following that 24-week period, if they are responding to the drug, they will have the opportunity to stay on the drug. So we will enroll 3 patients, again in the same way, where the patients will be started sequentially after they've reached the first safety milestone. So we'll start a patient. 2 weeks later, if there's no adverse events, we'll start the second patient, then the third patient. Once we get through the 10-milligram cohort, and all of those patients have been enrolled, assuming that there are no infusion-associated reaction problems or any other safety concerns, we will then seek to enroll 20 patients in the 20-milligram per kilogram group. So all of this is going to take more than a year to complete, assuming that we can have all of our patients lined up and ready to go as each one reaches the milestone. The primary endpoints, again, are safety and an infusion-associated antibody response. Secondary endpoints, pharmacokinetics, will again be looking at measuring GAA levels. We'll look at glycogen in the muscles. We're going to do a few more respiratory studies, mean expiratory pressure, mean inspiratory pressure, which are, again, ways to evaluate lung function. And then the 6-minute walk test, which is being able to walk 130 feet in 6 minutes. And then a QMT, which is a different test, and that's a quantitative muscle testing test in which we actually measure changes in muscle function. So I know that in many Pompe patients, they have difficulty with their quadricep muscles. And so that would be one of the things we would measure. Has your ability improved? Do you have more muscle function as a result of being in the trial? We are expanding the age of patients from 13 to 65 years. We are still looking for people with 75%, less than 75% of the lower normal GAA level and more than 40% of forced vital capacity. They must be naive to GAA enzyme replacement therapy. And the reasons for that, I know we've had a lot of questioning about that, is that once you've been exposed to enzyme replacement therapy, your body does form some antibodies. And even though it may be months or years since you've received any enzyme replacement therapy, those antibodies still exist in you, and the minute you start putting in more enzyme replacement therapy, it pushes those levels back up. Well, by doing that, we run the risk of getting some very misleading data because people may have very high responses. They may have responses in a different manner than people who are naive to the drug. There's also the possibility that there will be more infusion-associated reactions, and that could force us to keep the dosing at a lower level than we would like to, because obviously if we can put more drug into patients, we would expect more beneficial effects. And it could even force us to stop the study, which we certainly wouldn't want to do. And then the third reason is that if you've already been on enzyme replacement therapy, you may have received some or all of the benefit that you could possibly get from it, in which case we would not be able to tell if the drug was working. So by putting it into totally naive patients, it's like starting with a clean slate. You should never have had any infusion-associated reactions. You shouldn't have any antibodies, and we should be able to see the maximum benefit of the drug, which is helpful to us, obviously, from getting the drug approved, but also helpful to you as patients to know what you can expect from the drug. So at this point, I have concluded the formal presentation, and we can open it up for questions.
Ladies and gentlemen, at this time we will conduct a question and answer session. If you'd like to ask a question, please press 7# on your phone now, and you will be placed in the queue in the order received. Please listen for your name or location to be announced, and be prepared to ask your question when prompted. You may also continue to send chat questions using the chat window to the right of the presentation.
Okay, in looking down the list of questions, I think the first one is from Alison Locke, who is, I believe, in New Zealand, and she is asking, have all the sites been chosen for the upcoming study? Allison, they have not all been formally chosen, but as I mentioned, they will be in the U.S., the UK, and the EU. And the reason we're doing that is that we are trying to use experienced investigators who are used to doing these kinds of studies and will be able to get them done for us the most effectively and in the least amount of time. John, can you see the next question?
We have a few audio questions if you'd like me to bring those up in the meantime.
All right.
Our first question comes from Jim Gantz.
Hi, how are you? I'm wondering if you've considered doing in a approximately 30 late onset study, if you considered a group that aren't naive to GAA-ERT but have been off of Myozyme for 6 months or more, only because could you keep them separate and see how the drug affects them? Is there some reason you couldn't do just a category of them so they don't influence the group that's naive?
John, I'm going to give that to you.
John, are you still with us?
I'm switching him over to the presenter now.
Well, it doesn't matter. He should have
Okay.
audio contact unless
Okay.
he self.
Sorry, I was on mute, guys. Can you hear me now?
Yeah.
Yes, you're fine.
I apologize. What I was going to say, well, in fact, what I was saying, but to no one, was that that's a very good question, and we have considered that. I can tell you that most certainly we will, during the clinical process, at some point in time, be testing this compound in non-naive patients, and conceivably it could happen in this study in the high dose group. But at the moment,
he broke up.
Sorry, you still hear me?
Barely. You broke up at the end there.
Well, what I was saying is we will most definitely do that at some point in this test naive patients. Or not is a different question. And it's not impossible that it could happen in this study, as you said, taking a group of patients, perhaps at the high dose group where we anticipate as many as 20 patients being enrolled. That's something that we think is an idea that has some appeal, but at the moment that's not part of the clinical design.
Okay, thank you, Jed.
You're welcome.
Our next question comes from George Fox.
Hello, can you hear me?
Yes.
Okay, I have a couple different questions. Thanks for taking my call and thanks for having the webinar. This is really great. The first question has to do with autophagosomes. I'm sure you're familiar with the autophagic buildups that Nina Rabin has presented before on similar webinars. And your GILT technology interacting with autophagosomes as opposed to not necessarily glycogen clearance so much, but how does it work to alleviate some of the problems associated with autophagic buildup? And along those same lines, have you seen any positive strength gains versus, glycogen clearance? We've seen a lot of glycogen clearance in the past with other approaches, but have you tested to see what kind of strength gains you've gotten on animals or other?
Yeah, two very good questions, George. Thank you for them. The answer to your second question is short, and the answer is no. We've not done that yet. We will be doing additional animal studies as this program advances at BioMarin. Your first question, I think that's a very interesting question. I would say that number one, the autophagic buildup is a secondary pathology to the accumulation of glycogen in the lysosome. So it does have as its root cause still glycogen buildup. Now, whether you can reverse the pathology associated with some of the sorts of autophagic vacuoles that Nina Rabin has described is another question. In that slide at the very beginning that I showed you, I think it was, the second or third slide, there was, some cartoons describing the sort of pathology that had been observed by the genzyme pathologists. And it seems clear that there are levels of pathology that cannot be reversed. I think that is true for this compound or for any other compound. I mean, there will be some levels of damage that cannot be undone. However, I do think that in some of Nina Rabin's papers, she talks about misdirection or localization of GAA molecules due to this autophagic pathway being prominent, and so there's inefficient delivery of the enzyme to the lysosome. I think at least, whenever it's a numbers game, this compound will be delivered more efficiently. And so I think there is reason for optimism that not that everybody who is afflicted with Pompe disease will have all of the clinical manifestations reversed, but that one can not only stabilize the disease but reverse a more significant amount of the pathology.
Sure, yeah, that makes perfect sense that if you can sort of intercept the disease before it gets to that, critical threshold where it becomes more of an autophagic problem as opposed to a glycogen problem, that makes great sense. So I guess my last question has to do, and you may hear it again from other folks, is the possibility of getting BMN-701 into children, obviously younger than 13 years old, but more or less we call them infants. But the kids with that have cardiology or have heart problems associated with Pompe disease. I know it's kind of a difficult thing to do. Just sort of be advised, and I know that Myozyme was initially approved using kids. It was much more difficult to get it approved in the adult population versus it was pretty cut and dry in the kid population, that if you could save their lives, then you could approve the drug. And theoretically, if you could do this with the BMN-701 with lower dose, let's say whatever the lower dose may be, and you have the same sort of robust cardiac response, and I know you guys have thought about that, but we certainly would like to see this product get into kids if that's possible.
Well, I think again that's an excellent point, George. At ZyStor, we saw that as a viable clinical pathway. The FDA, I should tell you, was unwilling to let us do a first-in-human trial in infant onset patients because, rightly so, there's an ethical issue where there is a life-saving drug approved. You can't justify giving an experimental therapy to such patients. So it's quite clear that the beginning of our clinical pathway has to be in late-onset patients. It's not as clear what will come next, but I think you make very good points.
Do you see any way, because it does present quite a hurdle to get into kids, because most of the kids will obviously be directed toward the approved Myozyme, so it could be kind of a difficult pathway. Certainly, we've had our son has been off of drug for up to a month before. and if one of the inclusion criteria was to be either naive to GAA or a month off, I would certainly, in my case, certainly be willing to do that. To once the drug has been tried in adults, I would certainly be willing to try something that could show more of a response. Because I think it's interesting if you have, you say that you had this, that you've had myozyme in people before, that it might, and I guess there's a lot of variables out there, but it might skew the results, but it also may establish a new baseline, sort of a myozyme baseline, whereas when you present the BMN-701, now you have this new, you would have a definite either increase or decrease maybe in strength or whatever. So I guess there's a lot of different ways to look at it. But
well, again, all very good points. I think all of these things have been and are continuing to be discussed. I think the very first thing we need to do is see and demonstrate that the drug is safe, okay? That it's safe, number one, and number two, to get a sense of whether it's likely to represent an improvement compared to myozyme, okay? If we can show that, then I think a lot of clinical pathways are possible. And so, it's a little premature to start, designing the pivotal phase 3 studies before we have data from this study. But I do appreciate what you've said, and believe me, we spent a lot of time thinking about, possible clinical pathways, and we will continue to do so.
Well, I appreciate that, and thank you very much for the answers, and good luck with everything.
Thank you.
Thank you.
John, can you see the chat questions?
It's a question about...
You're breaking up, John. John?
Why don't you read the questions to him and let him respond?
All right. John, there's a question from Allison Locke. Is there anywhere in the Southern Hemisphere that we're able to partake in this study? And I would say at this point, not yet, Allison. I mentioned that we would do it in the U.S., the UK, and the EU first. After we've gotten there, then we'll be expanding to the further parts of the world. I don't have a timeline for that. I wish I did. Okay, John, I'm going to read this question to you. It's from Monty Frost. You have talked about the benefits of BMN-701 for removing glycogen. This sounds great, but what about the effects of it, if any, of reducing the problem? We already answered that autophagy question. Next question is, when does the trial, if the trial goes as expected, when could the rest of the late onset patients expect to receive the drug commercially? I think we may have lost John altogether, guys. It's 2 or 3 years away. This is a phase 1, phase 2 trial. After this data is analyzed, we'll have to go back to the FDA with our proposal for the phase 3. The trial will have to be done. The data will have to be analyzed. Then we will submit the new drug application, and the usual approval process is somewhere 6 to 9 months. So that probably takes us out to 2013. Monique's question about the enhanced delivery system, does it mean the possibility of shorter infusion times or smaller doses?
Barbara, I'm back, by the way.
Oh, good.
My phone ran out of juice. Yes, I can see.
Can you see Monique's question?
I can see Monique's question.
All right. Why don't you answer that?
Thank you, and I'm very sorry my phone died on me. Okay, so with this enhanced delivery system, does that mean the possibility of shorter infusion times and or smaller doses, which might equal a lower cost? So I think the short answer is yes. It could mean lower infusion times, and it could mean smaller doses, and it could mean longer intervals between infusions. could mean all of those things. And it's also possible that, if depending on what turns out to be the most efficacious dose, it could be that the optimum therapeutic benefit occurs at the 20 mg per kg dose, which, if we were to extrapolate from the animal data, that might be the dose that gives you the equivalent, say, of 100 mg per kg myozyme. So all of those things are possibilities that will have to be explored during the clinical process.
Okay, can you jump on to the next Monique question too, Jon?
Okay, so Monique's next question is, what are you using to measure glycogen levels? Muscle biopsy, MRI, other? Yes and yes. And I should say that I think many of you are well aware that at least in adult onset patients, the argument has been advanced that it could be challenging to generate muscle biopsy samples that give representative readouts of the glycogen content in the muscle overall because of the heterogeneous nature of muscle tissue from patients afflicted with Pompe disease. So we are not only going to do needle muscle biopsies, but also MRI to measure muscle volume and magnetic resonance spectroscopy to try and quantify glycogen in a non-invasive fashion. These procedures, by the way, I should point out, will likely be optional procedures in the amended clinical design.
Can you take Jared's question too?
Jared's question, what is the likelihood of signaling through IGF-1 or IGF-2 receptors using this compound? And is this the cause of the hypoglycemia? So first of all, the IGF-2 receptor, which is also the mannose-6-phosphate receptor, generally is believed not to result in any signaling pathways being activated, although there have been one or two reports in the literature of such. I think it's likely that one can discount those. As for the hypoglycemia that was observed in the animal study, we haven't proven it, but I think that a viable hypothesis is that you are seeing receptor crosstalk. That is, that the tag at very, very high concentration. So again, we're talking about 125 milligrams per kilogram given in an IV bolus. That means all of it at one time instead of being infused over many hours. So at these very, very high concentrations, I think you possibly can see some transient signaling related to interaction with the IGF-1 or possibly insulin receptors. So this is something that we've discussed with the FDA. We have, and the FDA has become comfortable, I think, with the idea that it's not likely to appear at the doses that we are using in this clinical study, and B, that this is something that is easily controlled, particularly by making sure patients are fed or that they're provided with fruit juice and so on. And then finally, we're going to be monitoring blood sugar for patients during and after infusion. So we will certainly pay careful attention to that. I might add, Jared, by the way, that not all signaling through the IGF-1 receptor pathway is necessarily bad. For example, we think this possibility of receptor redistribution, which is likely to go through the IGF-1 receptor, might actually improve delivery a small amount or perhaps a significant amount. And I guess the final point I would make is, in the animal tox study that was enabling for the IND, we saw no evidence of any sort of chronic effect of signaling through the IGF-1 receptor. So, there are a lot of very straightforward things that you might expect to see if you were getting chronic effects because, for example, IGF-1 has been approved as a drug, and in the case of the animal tox work with IGF-1, you see marked increases in organ weights and in overall body mass. And we don't see any of that, nor do we see any chronic hypoglycemia in the animals in the study. So we think that while it, we did see some signs that there could be transient signaling through these receptors, we don't see any chronic effect. And so, I think that's sort of a summary of where we are. There is a CHO cell. Oh, Joe has answered his own question. Okay.
Okay, and patients who have used Myozyme or Lumazyme would be eligible to use our drug at some point in the future once it's approved.
Absolutely, I would think so. And I think, as I said, at some point during the clinical pathway, we're probably going to want to explicitly test this compound in patients who have been on Myozyme. It's just our feeling was that in the first in-human study to get a really clean picture of safety and a glimpse of efficacy that would only complicate the signals. Have we missed any questions?
I don't think so. Oh, yes, Joe Boric had a question. It says, ZyStor uses a proprietary cell line. Is it embryonic?
Well, he's answered it below.
That's right.
The CHO cell line.
The CHO cell. No, I think that's it. And I think we're out of time, too.
Well, in that case, I want to thank everybody for joining in on this call. It's always just exhilarating to talk to the Pompe patient community, and it's really remarkable to me how well informed as a group you guys are, and I really do appreciate the interaction. I'm sure I'm speaking for Barbara as well, and in fact, entire Pompe team at BioMarin when we say that we thank you for your attention.
Thank you very much.
This concludes today's AMDA webinar. Thank you for participating.